Okay, good morning, everyone. Welcome to the June ALK2 meeting. We have quite a bit on the agenda today. So let me just pull the agenda up. How can I get rid of this?
Just here,
this one?
Yep. Move it around. Just go on now. Yeah. Great.
Okay, so with regards to today's agenda, I'm going to give again just a brief intro just to remind people where we are, where the project is, and I'm also hoping to give some updates of some of the data that we have collected over the past month. I wanted to remind everybody that the meeting is being recorded, so just be aware of that. So I'll give an introduction to the project for today. I mean, we have a lot of things to discuss. Then Alex and Jean would give an update on some of the cell-based and nanobit data set. Then we're going to get an update from Jerome about some of the combination studies we started looking at. Sue would give her update on some of the chemistry on the backup series. Then Diva is going to give an update on some of this around the pyridine series that we're looking at. And interestingly, so Owen had organized this meeting with clinicians so we can start getting an idea of strategically what we want to do with a project like this or a target like this as we enter the clinic and get some feedback from clinicians. And we got some really exciting feedback from those discussions. So Owen and Arish have summarized that. And so we'll give you an update of some of the outcome from that meeting. And at the end, if there's any burning issues, then we can continue the discussion.
We have an hour of discussion in next step, so we'll see if we can cut that down a bit.
So usually we should be finished before that. All right. So, okay, what I thought I'll start off with is some of the actionables that we had going from our last meeting. So, you know, one of the exciting things from last month was we had gotten the FOP data and we had done it at just 100 mg per kg at one dose. So we had some discussions as to whether or not we should really look at another dose, and we agree that maybe yes, we should look at a lower dose to get a better sense of a dose response. And so Eileen agreed to look at the 50 mg dose. We looked at the 100 mg dose once a day. She agreed to look at the 50 mg dose, and she is doing that experiment as we speak. We haven't gotten any full update from her yet, but as soon as we do, we'll share that data set. The other thing that Jerome is going to start discussing, something we have talked about at length, is the combination strategy with ALK2, and so you're going to see some of the first glimpse of some of that combination data. I mean, we looked at a number of different kinds of combinations. It's a big experiment, so we have done some of the compounds so far. So I have like partial completion. We have done, you know, about half of the compounds we wanted to look at. We probably have another set of experiments to do. Again, for me, something that's really dear to me with this project is really establishing, you know, cell lines that we are convinced are dependent on ALK2, so we can actually have DIPG models, kind of very similar to FOP, where we know ALK2 is the driver. So if you can identify any patient-derived line that's a real ALK2 driver and we can engraph it, that would actually be ideal. And so I think it's going to be a continued effort. You know, some of these cell lines, not all of them are ALK2 dependent because they have other lesions, but I think, you know, that doesn't prevent us from actually trying to find a cell line that we can actually use as a true ALK2 driven model in the DIPG. The other thing, again, now, one of the kind of the key phase of the project is identifying, you know, whether or not 2009 is actually going to show efficacy in vivo in some of the DIPG models that we have available. And so a number of us have discussed about doing animal model studies with 2009, which is available. So we are in discussion with Chris Jones, Angel, who have also expressed an interest. Now, Jerome is also looking at some of the cell lines, and Julia from Charles River was also dabbling in some of the cell lines. So I have here a lot of these are being planned. It would be nice. I know a lot of these models take quite a bit of time to develop, and so a study is going to take about 4 to 5 months. And since if you're hoping to have data by sometime in November, I think we have to seriously start thinking about getting some of these experiments on the way. And then the last 2 points I have here for actionables were, today we said we were going to start focusing on a short list of compounds that we can pick from the lead series. You know, the efforts aren't going to be complete by any means, but we just want to start picking a few compounds that we think we can select to declare as a preclinical lead by November. And then hopefully we're going to also have some backup compounds from some of the backup series. You know, Sue and her group is also working on the blueprint scaffold. So we're hoping to have a lead compound and some representative backup compounds. So a lot of this effort is still ongoing. Okay. What I thought I'll do is kind of remind people again that, you know, we are really after therapies to treat DIPG, which is really in need, but there are no good models currently, and we're trying to identify models that are suitable for the DIPG disease. So in the interim, we have chosen to use FOP as an animal model where we know the ALK2 activating mutation is causal. And so the ILA model, which is an inducible model, is a good model. We've actually put 2009 in that model and started getting hints of activity, convincing us that we're actually engaging the target and getting the required pharmacology out of that. But more and more recently, there are a number of ALK2 compounds that have been published. I mean, thanks to Sue identified a Novartis patent. And I thought it was interesting, some of the assays that they're putting these ALK2 compounds through. And I kind of highlight one of them here. And so it's called the turpentine oil-induced model in rat. And so that's to treat a disease that they call anemia of chronic disease. Things like cancer and inflammation tend to be associated with anemia that leads to a lot of fatigue and stuff. So in order to improve like patient outcomes and stuff, people believe that these types of anemia need to be treated. And it's interestingly that the anemia of chronic disease seem to be ALK2 driven. So ALK2, when it gets activated, activates MADs and leads to the production of this peptide hormone called hepcidin, which actually regulates iron levels. And so if your iron levels are actually disrupted, sometimes that can lead to a lot of fatigue and stuff. So that's a model people are actually starting to look at. And it's interesting because the FOP, which we think is a good animal model for the disease, it's quite involved. You've got to do the induction and then measure all the bones and stuff. And it would be nice to get a nice quick PD model where we can assess compounds in vivo to see which one is good or not. And I'm starting to think that this might be a model that could serve that role. I mean, it would be nice to hear feedback from other people. So I'm going to do a little summary of some of the data that comes out of these type of experiments. So, okay, so just remember that this whole ALK2-driven hepcidin expression, it increases serum levels of this peptide hormone, which is easily measured by mass spec. So it's kind of like a PD, I mean, you know, just like when you're doing a PK and you take blood samples to measure the compound, you can take blood samples to measure this hormone because it's driven by ALK2. And I just show the schematic here that when the receptor, the bone morphogenic protein receptor, which is ALK2 here, gets activated by BMP, it activates MAD and leads to induction of this particular hormone. So it was interesting here. So here's what the model looks like. I mean, like I say, it's a typical PD model. So basically, it can be done in rat and mouse. So either you take a rat, you know, typically you got a vehicle control and you can measure using mass spec. Okay, you can measure the level of hepcidin in serum. Then what you do, you treat the animal with turpentine oil. You inject sub-Q. So you could see here, if you inject about, sub-Q about 1 ml per kg of turpentine oil, you get an induction of hepcidin, and you could see there's a time course to it. So the clear bars is 6 hours after injection of turpentine, Okay, you see about what, a twofold improvement in turpentine levels. 24 hours later, it's up to about almost fourfold. If you now treat with and this is the ALK inhibitor down here in the corner, you could see that's in your structure from Novartis that supervised to us. 3 mg per kg of that compound, you could see you get a decrease at 6 hours. Okay, but at 24 hours, that low dose wasn't enough to overcome the effect. However, if you go at the higher dose at 24 hours, you could see this nice decrease at 24 hours, or you could see very, very nice decrease at 6 hours with the lower dose. So it could be a nice PD model we can use to really rank compounds, I think. If we get a short list of about 3 or 4 compounds, we can see how well they are behaving in vivo by just looking at this quick PD model. So it's something we could start thinking about. And it's all dosing the compound, and at 6 hours later, just like we take samples for PK, we can just take samples and measure the peptide hormone by mass spec. So I mean, that's something we can discuss with Ahmed.
So I just quickly, I mean, this is really interesting because it's obviously a shorter and more could be a cost-effective way of evaluating it. Have you seen this in any other patents and any other use? I know you did a lot of research and found it in this patent.
Yeah.
Have you seen it anywhere else, and how well validated it is?
Yeah, it's used routinely. Like, I mean, there's another mechanism. IL-6 can also do the same thing. And so people have used it. Some of those JAK inhibitors,
right,
you can actually use them in PD models like that. But what's nice, Tolero, which is in the clinic looking at, DIPG with their compound, the way they evaluated this was also using the same model. So Tolero have shown activity. The Tolero compound, which is an ALK2 inhibitor in the clinic, which is TP0184, they have also shown activity in this model. That's what they did in the patents that they have. So it's something people are using because, as I say, it's not as involved as the FOP. I mean, it's quick, it's a nice PD model, you can know you're on target, and the pharmacology for that is well borne out. So, I thought it's something we can use to either do compound ranking just to see how well the ligands of our lead compounds are behaving in vivo. It's a good in vivo surrogate of ALK2. Yeah?
It's Alex here. Just a comment on this. It's very interesting. The only thing is that this pathway is dependent on both ALK2 and ALK3, and particularly ALK3. So it would really rank your compounds to how well they bind both ALK2 and ALK3 together. So if you had a super selective ALK2 inhibitor, it would actually appear worse in this readout. You would have to be careful how you read that.
Okay.
Caveat, yeah.
Okay, that's an important point.
So blocking this pathway requires hitting both ALK2 and ALK3, or either?
I think both. I don't know the percentages, whether it's like 2/3 ALK3 is important and 1/3 ALK2, but I think both of them contribute.
Yeah, I mean, people have the knockdown of both, and they both contribute.
Method, do you happen to know what the profile is of the Tolero compound versus ALK3?
ALK3, yeah, a lot of that profiling for the Tolero is not known. So in the patent, they claim that it hits JAK2 and ALK2. So those are the two targets that they disclose in the patent.
Okay.
Yeah. Yeah, but a lot of biology, like Alex just mentioned, have gone into showing the contributions of the different ALKs to that pharmacology. So it's a nice little PD readout for that BMP signaling. And like as I said, we can kind of tweak out, keep an eye on, like he said, maybe we should, when we broadly profile, we should keep an eye out on the ALK3 activity also. I thought it's a nice PD model for the target family. And so it's something we can potentially use. It's quick, so we could see how the compounds are behaving in vivo. Okay. So what I wanted to start getting into now is how are some of the other compounds looking in our backup strategy relative to 2009? So again, wanted to remind people, so 2009, we like it very much. It's one of the lead compounds. You have scaled it up. You have tons of materials to actually do a number of in vivo studies. The only thing that we're lingering for us is these 2 points highlighted here in yellow, is we want to improve the half-life a little bit. And then there's some discussions around the issue of the herd. Okay, so you're going to see with some of the efforts going forward with the backups, I mean, these are some of the features we're trying to improve on. But before we get into that, I mean, also, we are still trying to identify a cell line that we can actually use to determine, you know, efficacy in DIPG models. And so a number of groups have been looking at proliferation assays and stuff. So, you know, we're going to get some update from Zhongfu of how some of these compounds are performing. I think Chris Jones has taken 209 and has evaluated it in some of his cell lines. But what was interesting, Angle have also taken 209 and evaluated it. And it was interesting, he got some updates. I mean, I don't know if Angel is on the line, but I summarized his data. And, you know, this is always interesting. When Angle look at 2009 and another compound, we had 2163 that was quite active. In the 3-day assay, you realize he got only partial inhibition. Okay, and so we kind of recommended that why don't you run the assay for a little longer to see, and you could see if you let the assay go for 5 to 7 days like Zhongfu and those guys do, then you start getting more robust inhibition.
Well, it goes lower, right?
Yeah.
But the IC50,
yes,
which
yeah,
is
yeah.
important. I mean, so yeah, that makes sense.
Yeah, yeah. So now he's now interested in really putting it in. He was questioning whether or not he should even go in vivo based on the 3-day data. But now he's seen the 5-day data, he's now convinced it might be worth taking a shot. So that's important because, like as I said, depending on cell lines and stuff, and the cell density and the growth rate, sometimes going a little longer, you can actually really map out the phenotype. Because 3-day assay, it kind of reminds me, that's an assay people used to use in the old days to look at cytotoxics and say in 3 days. But if you're not looking at real toxic agents, sometimes it's like a lot of epigenetic targets. You got to go out a couple more days to really see the profile. So this was quite encouraging to see. So that was some data generated by Angle. I'm sharing with the team. Another thing we have done, and we've talked a lot about it again, because a lot of these DIPG lines are patient-derived, and you know, we are doing orthotopic models because, you know, they may not all go well when you're actually taking them out of the environment. A lot of lentiviral inspection techniques can also be a challenge. So one of the things we kept talking about was designing a PROTAC of 2009. And one of the challenges of PROTAC sometimes is when you try to add PROTAC linkers onto them, sometimes you lose the pharmacology you desire. But it was nice. We had a lot of 2009, so we thought we'll just add, you know, a representative PROTAC onto it. And we were happy to find that the ALK2 biology was still retained in terms of the biochemical assay. And the PROTAC appeared to be quite selective over ALK5. So that was encouraging. I think Zhongfu have actually put it in cells. Now, typically these PROTACs, they aren't as permeable as the, you know, the actual ligand. So there was quite a shift. I think it was about 2 to 3 micromolar in the nanobreath assay, but enough of it gets in, because if you're causing degradation, I mean, this is a mechanism, as long as the compound gets into the cell and degrades the protein, then you should see a phenotype. So I mean, so I'm hoping that the next step is we can actually take this tool compound and start characterizing some of the DIPG lines to see which one might be truly dependent on ALK2.
Quick question about that. I think probably this is something that's been discussed before, but how good these things are at getting in the brain? Usually, is this something that people have looked at, or? Well,
not brain. So that was always one of the challenges. When you put PROTACs on, the PK becomes a challenge, but now people are meeting those challenges for other disease that are peripheral. I mean, I haven't seen anybody gone after brain disease with a PROTAC, but now just the typical like prostate cancer and stuff, you just got to get the compound into the blood. Some people come up with formulation to get at least plasma exposure and starting to show efficacy of these PROTACs, because I mean, if the target is gone, then hopefully you see your phenotype. And so, I mean, you know, there's some groundbreaking stuff happening here. But I mean, this is more for in vitro. We can characterize them in vitro.
Yeah.
I haven't thought about getting a PROTAC into the brain. I haven't that far ahead yet. But it'd be nice to just be able to characterize the DIPG lines that we have available. If a small molecule can chemically determine which ones are ALK2 dependent and which ones are not, then that would be ideal for us to develop models that we know are going to be driven by ALK2. So it's a kind of a tool we have. I mean, we still have a little bit more work to do in terms of validating that we're degrading ALK2 in cells. So that experiment is the next key step that we need to do.
I'm sorry to walk this back, but what's the problem we're trying to solve by doing this? I mean, it's interesting, but what's the problem that we're having that we're trying to solve?
So when I joined the project, we had all these DIPG lines. We know they have ALK2 mutation, but there was the question, are those cell lines actually dependent on ALK2? Because they have a lot of other lesions. We have the histone mutations, some of them have PI3 kinase. So it'd be nice, that's like in FOP, where we know the causal effect is a mutation in an ALK2, and that's the driving event. So if I get an ALK2 inhibitor, you should inhibit the disease.
Right.
That's not as clear-cut in DIPG because there's a lot of things involved. So I think to develop a clear ALK2-driven DIPG model, it would be good to know which DIPG cell line is truly dependent on ALK2, so that I know confidently when I put an ALK2 inhibitor inside there, it's going to always work. So
you want to differentiate the kinase activity from having the protein there at all.
Exactly. Exactly. So that was kind of the line of thinking. So as we get, and like as I said, these cell lines are very finicky. You take them out of the pan, and you could see these guys are trying to grow them orthotopically. So this might be a nice, easy way to just figure out, okay, of all these DIPG patient samples that we get, how can we quickly determine which of them are ALK2 dependent?
We should just also CRISPR out HDVR1, ALK2 in those cells.
Yeah.
And just establish those cell lines stably have been like knockout for
exactly.
and beyond. When I was in San Francisco, I talked to Nada Chabado. She said that either her lab or a lab that she knows has done that.
Mm-hmm.
And I actually talked to her after, but somehow there was not a proper follow-up. But
yes.
Yeah, so I think that would be a great experiment to do.
Yeah, yeah. And it's also it's a chemical way of knocking out ALK2. CRISPR is also good, but those take time and stuff. So it's an alternative tool we can help to just firm up some of that.
But first you got to determine whether it actually works by degrading.
Yes, yes. So that's right. So my point now is the ALK2 degradation still needs to be determined. So we know we hit the target. Well, that's why I'm throwing out the question. We're not the biologists doing it.
But at this point now, if you want to take it further forward, the problem of permeability crossing the BBB, those things will come in play. But Nathan is not proposing that at this point. He's just trying to see if we can find the ALK2 dependent line using ALK2.
Yeah.
But it's also going to be an ALK2 inhibitor. So
yes.
So it's when you take it into cell lines without knowing that it's degrading the protein, you don't know whether the phenotype is just the ALK2 inhibition.
Yes.
Whether it has anything to do with protein degradation.
Yes, so we still need to do protein I'm proposing that we still need to do protein degradation before we go to the next step. Yeah, yeah.
That would be the next step.
Yeah, it's not too bad to do. Just got to commit and have somebody. So I don't have anybody on the team. So I have about 20 mg of it available. You don't need much to do. All you need incubate your compound, wait a couple of hours, do some Western blots for ALK2, and you can see the protein disappear at different doses. If that's the case, then we might have a nice chemical tool to just quickly assess ALK2 dependence. Okay, so that's kind of the mindset as we try and develop tools. The other thing I wanted to kind of update the team on is, so we had done this FOP model with Eileen, with the notion that can actually serve as a surrogate for the DIPG disease. And so in that experiment, we wanted to know what the brain exposure was. So we had collected plasma and brains from that experiment, and I'm happy to report that in that experiment, quite a bit of the compound did get into the brain. We had a brain plasma ratio of close to 1. So that gives us confidence that 2009 does indeed get into the brain. And so if ALK2 is driving any form of DIPG, then 2009 should actually work if it gets into the brain. So that was important to note. With regard to backup compounds on 2009, 2 compounds that were identified that were quite interesting was 2117 and 2163. You can see the structural differences highlighted here in color. But the key that people have to keep in mind is that even though these compounds, so here's the target product profile on the right. What we like about 200117 is that you had better selectivity over ALK5. You could see it's 113-fold selective. It's about 2-fold, a little bit more potent than 2009. However, it's still plagued with hERG issues, so we didn't solve the hERG problem with that one, but we did improve the half-life in animals. Similarly for 2143, again, we got better selectivity. I mean, and another feature for that compound is its volume of distribution, which might be good if it gets into the brain, and we know it already penetrates the brain based on brain plasma studies that we have done. So we thought these were two interesting backups, but again, the hERG was still an issue for these compounds. That's why we have actually gone and devote a very, very strong effort to overcome the hERG. And so what I'm giving you now is some of the update of some of the hERG compounds that we profiled. We have profiled about 16 compounds, and I'm trying to give you a snapshot here. So the first column in yellow is, that's the hERG column. So I took the activity. So these compounds all had variations in the amine portion that usually contributes to a lot of the hERG activity. So we want to see if there's any SAR related to that. So we had run about 16 compounds. So this is a set of the first 5. And I've combined the hERG numbers with some of the additional profile that they have, some permeability numbers here at the end, and then the selectivity over ALK2 biochemically and also in the nanobread assay. And so you could see, you know, for the first 5 compounds on this table, we also reran in the hERG assay 2009. And so the first time we ran it, we got about 8.2. This time we got about 7.2. So, you know, 2009 is still consistent about, you know, 6 to 10 micromolar hERG activity. However, you could see something like 004, some of the variations on the amine portion doesn't take care of the hERG at all. It's interesting, the compound at the bottom, 853, when we homologate the piperazine out, you could see you improve the hERG profile a bit. Target cutoff I have is 15 micromolar in my criteria chart, so that's why that's green. But the thing that happens, that improvement, you actually see a drop in the cell-based activity. It was now almost 108 nanomolar in cells. And in addition to that, that compound starts becoming very metabolically labile in mouse. And so that's usually one of the challenges. You're improving one, but now you're destroying some of the other properties. Okay, as we continue down the list, you had these 2 compounds at the top. They're very, very similar. Again, they both still have hERG. You could see the changes on the other left-hand side of the molecule. So we still had that hERG. There was no improvement over 2009, so we didn't see any huge benefit there. However, there's this other isomer of one of the backup compounds, okay, 2214, that still had, you know, similar hERG activity to 2009. Overall, its profile is very, very good. It's structurally distinct. Its molecular weight is smaller than 2009. Hopefully, it might have better brain properties. So we thought it's an interesting candidate to consider. That's why I have it highlighted here. It's interesting. You could see that change on the right with this dimethylamine. If you take the cyano version of 2009 and you put those, you could see now you improve the potency at hERG. This one is like less than 1 nanomolar at hERG. So that's not the direction you want to go into. Okay, so that was interesting to see. The last compound in that table, 806, was interesting. So that compound now, the difference is now we have gone from the left-hand side where we are modifying the amine to now the right-hand side where the trimethoxy group is, and we're replacing the central group with an amide. And that's where we see a lot of huge change in the hERG profile. That was a key finding for us. So now you could see now you're adjusting the hERG. These compounds are now greater than 30 micromolar in hERG. But the problem with that compound is its permeability have now gotten really worse, and the selectivity over ALK5 is lost. So as excited as we were about that hERG profile, I mean, some of the other features actually got destroyed. As you continue down, you know, the first compound in this column, you could see we fixed the hERG by attenuating the basicity of that compound, but then you could see we destroy a lot of the other nice properties for that. So that wasn't beneficial. But the other 3 compounds here are really, really exciting for us. Again, they're amides, themes, but now we've actually had these alkylated amines on the amide, and they all now have significantly improved hERG profile. And we didn't destroy too much of the other properties. We actually preserve a good portion of it. So these candidates are starting to look really, really interesting. Okay, so you're going to see in the next table what I've now summarized based on what we have. So in highlighted in yellow here is 2009. I've highlighted its biochemical ALK2 and its herd numbers. On top are 3 other compounds that we started in testing for other features. I told you about 2117 and 2143, and now that recent isomer. So you could see 2143 and 2214. The difference is the position of nitrogen in the bicyclic ring. And so we wanted to explore that a little bit more. But you could see the herd profiles are very similar, but 2214 is actually more potent than 2143. So we're going to see how that actually holds up. But at the bottom is now where we see some of the huge changes where that essentially my group now in the flow system, we have now improved the herd significantly while retaining a lot of the ALK2 potency. So we're currently doing, we're waiting on mouse PK, BBB. These have been sent out for SIP profiling and broad kinase selectivity to see how well, you know, some of these compounds are going to look overall. But we're hoping from the short list of the, you know, compounds from that pyridine lead series, we can actually identify at least 2 to 3 that's really have all of the features that we desire. Okay, so, and then this is just now I'm filling out the product profile relative to 2009. So as that data set comes in, you're going to probably see a lot of this. I'll send the data set when it comes. I mean, so I think within about a week and a half, we should probably get most of this data set pulled out so we can make a clear decision as to which compound we want to go forward with. And lastly, not to forget, you know, we still have a backup series considering the blueprint scaffold that CRL is working on. So 3078 and 3079 were 2 compounds. We've actually checked the permeability of those, and they were looking very good in CACO. So we're interested in evaluating these in PK and BBB to see how good they're going to be. One of the things Jeff had done before he left is we always wanted to know what the blueprint clinical compound was. We didn't know. Now, when the structure got revealed, we actually had it made. And so the synthesis of it was just complete. We now have 300 mg of it available. It's called Blueprint 785. That's going to serve as a good tool. It's in phase 1 from Blueprint for FOP. Now it's in hand, we're going to characterize it and see how potent it is and get all the additional data for it. So, you know what I mean, it's something we can use that information to just help characterize where these compounds are going. And lastly, I wanted to just remind people, I mean, the competitive landscape. When we started out, I mean, you know, a couple of compounds were in the field, but people are now starting to publish the FOP ligand. So I mentioned this Tolero company. It's a company from Salt Lake City. They were acquired by Sunimoto from Japan in 2017. So it's Sunimoto who's actually pushing these compounds into the clinic. So TP0184, we know is an ALK compound that's in phase 1. And when we were discussing with the clinicians, they did mention to us that this is a compound they're trying out in DIPG patients. So just to know that, that's a compound people are looking at. Now the company has 1 ALK2 patent. When I went to the patent is about JAK2 and ALK2. These were the only 2 ALK2 selective compound in the patent. So my hunch is the candidate is from one of these. So I've put the 2 compounds there. Hopefully when they disclose the structure, we'll see which one it is. And that's where they also had some in vivo data and stuff. A lot of the data was around compound 12. So I feel like a lot of the compound information around the lead compound might be the trimethoxy. It's interesting, that same trimethoxy moiety, you know, that's resident in our series too. So keep an eye out for that. I highlighted the blueprint compound that's here. Now there's a company called BioQuest, have also published a patent. They claim that they're in the clinic, preclinical, they have this compound called BCX9250. They haven't disclosed the structure, but I just show a representative structure from their patent for what it is. And lastly, Suscram sent this Novartis patent to us. This is one of the compounds that they disclosed in the patent that had a lot of data on the hepcidin model. So that's the data I just showed you. So just be aware of some of this competitive scope. Okay, and now lastly, with regards to timelines, I wouldn't belabor this point too much, but we are in the middle of June. We said that we're going to start looking at a short list of compounds. So we are close. We're starting to do that. The idea at the time was to at least get some efficacy with 2009 in a model and hopefully get one more compound in, but I think that's going to be tough, knowing how long the model takes and scaling up compounds. So I feel like for proof of principle, we might only get data from 20009, but I'm still hopeful if we get a really, really good candidate, we might be able to fast track it and get some data. So, so those are the two. And then again, we're hoping to declare actually a preclinical candidate from the pyridine series by November. But we still have compounds from the backup that we can also continue to look at. All right, so that's basically a wholesome overview of what's happening. If anyone have any burning questions, happy to answer them. If not, we go straight to Jung Fu's presentation.
Just as an aside, going back, I think, to the original grant, we're in great shape for this. I mean,
yes.
Just going through that list of compounds, I think there's a couple that we're not going to know until we do full preclinical next year.
Exactly.
Which one is truly better?
Yes.
There's a lot of talks that have to get done. There's a lot of understanding of molecule, et cetera.
Yes.
So I'm actually quite excited that I think we're going to, declare a couple leads.
Yes.
And it'll make for a very interesting project next year.
Yeah, I agree.
There's a lot of work and it's really exciting.
Yeah. Okay, so next up is Jung Fu, which is here. Jung Fu, you're up.
Hi, Metin. Is that? I think it's slide 3 of 3. Do you want to go back to slide 1?
Oops. Oh, sorry. Okay, there we go.
Okay, this is Alex. I'll just say a word about the crystallography. Last time Ellie had a few crystal plates set up that looked hopeful. We took some crystals to the Diamond synchrotron for data collection. Some data was collected, but when we analyzed the data sets, which is now much of the setup and data collection is now automated. So you have robots that put your crystal in the X-ray beamline. And we noted that for many of the crystals, the automatic setup had not centered the crystal in the beamline, and we think the crystals were deteriorated. They're probably not frozen by the nitrogen stream as well. So when we went back to recheck the crystals, they all seemed dead. So because the crystals are not properly in the beamline, they don't obviously diffract as well. So that we've lost a batch. So we can prioritize those again. Right now is a summer recess where the synchrotron has its routine maintenance. So there's going to be a delay on getting that, but if priorities change, do let us know, but that's just a shame. But we'll move on to Jung Fu, and he's got 3 slides on some of the cellular assays. Okay?
Okay, thank you. Thanks for that update.
Thanks, Matthew. So I'll just quickly read through, as you have so nicely summarized them, most of the results already. So like some of them, basically most of them deal with alkylation of solvent site group, and in the case of, for example, M4K2009 with solvent site modification, it drastically improved the ALK2 potency. And in terms of like selectivity towards ALK5, against ALK5 is not perfect, but they are quite potent. And also the addition of the pyrrolidine group is tolerated in the nitrile compound, and as well as the ones that you mentioned, the 2, 2, 3, 4, 3, 6, and 3, 8. They're pretty nice. And also for 2, 2, 3, 2, the alkylation is also improving the selectivity.
Yeah.
So can proceed to the next slide. And over here I've tested also the different synthesis batches, and the assay results are quite consistent. And actually, I haven't notified everyone with the result from Charles Brewer's lab, the M4K 30377 and 3089. I haven't gotten the ALK5 result yet, but those two are pretty potent towards ALK2 as well. and they are with the replacement of the two of the methoxy group, and which is quite interesting. And there are others. For example, the removal of the hinge binder methyl group severely diminished the selectivity. So we can move on to the next slide.
Okay.
So I've taken your suggestion with the most interesting compound, like 8 of them, and perform the assays in either DIPG cells with wild type ALK2 or ALK2 mutation, and also an unrelated glioblastoma cell line with wild type histone H3. Actually, it's having a different mutation of the histone, but a wild type ALK2. So none of the compounds were effective on the glioblastoma, as well as most of the DIPG cells with wild type ALK2. And as we've seen before, the DIPG 21 is basically responding as we expect. So I would think that it's probably completely reliant on ALK2.
Yeah.
So the EC50 is perfectly fitting to the potency of the compound, kind of. Yeah. But in terms of 007 as well as the number 4, the response is not as much. And over here, I would like to point out that the analog of the blueprint compound, again, they did not affect the viability of the DIPG 7 and 4, but they are working in the 21. So yeah, likely those two other DIPG cell lines, yeah, have some other.
And that's good. I'm glad you captured the reduction in the percentage, because like you said, maybe some of those blueprint compounds, they're actually weak. They're not coming down to the baseline. I hadn't captured the reduction percent, because those kind of could be partial curves, and maybe if you let them go, so they're just not as potent. So I'm glad you captured that. That's actually very important.
Thanks, Nathan.
Yeah. Well, I think it's becoming really evident that DIPG21 is one of the most sensitive lines to an ALK2 inhibitor. And so, unfortunately, that's one that doesn't engraft very well. So, I mean, if we get that one to engraft, I think also that would be a good line to study with the PROTAC, just to kind of validate that indeed, if we knock out the ALK2 protein, that's also going to be the most sensitive line. And if you can get that line engrafted, that would be the ideal preclinical DIPG model to be using to look for an ALK2 inhibitor. So that's kind of the dream. I mean, looking at ALK2 as a mechanism, if you can actually identify, a tumor model where, ALK2 is the driving event, and it becomes night and day. You put a compound in, it works. If it has ALK2, if it doesn't, you don't see anything.
Yeah. Yeah, I guess for the line 7 and 4, potentially they will work better in combination therapy.
Yeah. Okay, this is exciting. And then you're going to see some of the combination efforts from Zero in a minute. Okay, any other questions for Zhongfu? Yeah, this is fantastic. Thanks, Zhongfu. This is great. Okay, so we move on. There's no other questions to Zero. Zero's update. Zero is this one. Okay.
So we tested a few different possible combination of compounds on the IPG cell lines, and this is the 5 cell lines that we looked at, and this at the bottom is our experimental design. So it's a 4-day assay. So if you move to the next slide. So we selected compounds based on potential interesting targets, things that we know drive tumors or, could be interesting. So we have PDGFR inhibitors, HDAC inhibitors, epigenetic modulators, AKT, PI3 kinase. Some of these cell lines carry PI3 kinase mutations. And we just profiled them individually first in all the 5 cell lines. So the DIPG4, for some reason, didn't look great at all in those experiments. But we got some pretty decent data from at least 3 of the cell lines. So to test the combinations, we focused on the compounds that are highlighted with the red arrows on top for the first round, and we just did that on the DIPG007 and the DIPG36 cells because these ones can engraft in vivo. And we just talked about the DIPG21. At least in my hands, they don't xenograft. I don't know how you can make a cell that doesn't xenograft transplant. I mean, I don't know if that's even possible. I think that usually is just like, you derive the cell line, see if it works, and then if it doesn't, then it doesn't. But maybe there's a way, I don't know. Maybe you can engineer it in some way, I don't know. So if we go to the next slide. So I'm not, I don't do these assays routinely, so I've just been coached by Richard on interpreting these data, as he has all the algorithm to do that. And here we have a compound which, looked like it might have largely some additive effects, but I think Melvin's going to have additional comments about that. Essentially, what you're looking at on the top is the profile for the compounds individually, and then at the bar, like the third table that you have is how much more inhibition you see compared with what you would expect just by the additive effect of the compound. So the more red it is, the more likely there could be some synergy. And I mean, these are not, essentially here, the reason why I was thinking this might be more additive is because the numbers aren't that high, essentially, with this HDAC inhibitor. But if we go to the next slide, then we have this compound that may show what looked like maybe some mild synergy, because you do have some of these values there that, essentially you have more inhibition than you would expect just by summing up the effect of the 2 compounds individually. And with the red arrows, I pointed out on the dose response curve approximately where that happens. So it's kind of like at the shoulder of the dose response curve. And what Richard seemed to say was that this is kind of the pattern you would expect for synergy. Then you see also there's some green, which would suggest antagonism. And now this you have to take with a grain of salt, because this is where you get into the part where there's a lot of inhibition. And you often see drugs in combination when you reach the bottom that will show, this antagonism, but it's not real antagonism. It's just cells are dead at that point. And so what we want to do now is basically test a few other compounds. We have, if you remember from the one that we profiled all the compounds individually, we have like a few drugs, like especially the EED inhibitors that don't do anything by themselves. These might be interesting to look at in terms of synergy, because in those cases, if you see anything that's more than just M4K by itself, it indicates synergy. Also, just a kind of like a general comment is that additive is not bad. We like to think of synergy as something magical that's great, but additive can also be super great.
It's different mechanisms that add to the plus. I agree.
So that's basically where we're at now.
So we're going to do another round. That's good. And so what I've done, I've tagged on to, so Richard and I actually look at a lot of this data, and so I just added on to the bottom some different ways to look at the data. So this is kind of just to speak to what Jerome just said. This is a little summary table where you got the 2 cell lines here. I actually just picked 007 and 21.
I just want to clarify that this is 36, not 21.
36? Okay. But the data set was very similar in both.
It was similar.
Okay, and so one of the things we did, so on the next slide, one of the things we did is, this is an interesting way to look at synergism. And so what I've done here now is to take the EC50 of, let's say, the compound we're looking at in combination with 2009, and then look at how the EC50 of that compound changes as you increase the concentration of 2009. Now, I have highlighted here a red line to each of these graphs, and that's where the concentration gets like up to 5-10 micromolar, where you kind of start getting strange pharmacology. A lot of that is really non-pharmacological, but if you focus on, you know, exposure concentrations below 1 to 2 micromolar, you start seeing more meaningful pharmacology. And so it's interesting, if you look at 285, the PDGFR inhibitor, you could see over time as you're titrating the 2009, you see a slight improvement in the EC50 of 2009, and then it gets wacky at high concentration. So we believe that there is a slight hint of synergism with the 285. The other compound that's also showing some nice synergism, we think, is the HDAC inhibitor, the CNS penetrant HDAC inhibitor. So you could see, we see almost a twofold improvement here in pharmacologically meaningful concentration. We're quite excited about those two modes of, you know, potentiation. Now, the other two compounds, you could see the EC50 doesn't really change over time. It's this is a good indicator of additivism. Okay, so like the PI3 kinase mTOR inhibitor, 084, and so you could see the EC50 is not changing much. So it's just a different way to look at the data set. How is the EC50 of one changing with regards to the other? So we're going to do the other set of compounds and then come back and look at the data set wholesomely. But there might be something with PDGFR and HDAC, which, you know, the histones really play a part in, you know, driving some of the disease. So that might actually be a role that we are starting to see in some of these combination studies. Just to remind people, this is basically the data set of compounds that we picked. So we had some epigenetic inhibitors, EZH2E, we had picked the HDACs, some BMI inhibitors, and then a few representative kinases here. So
again, I just want to clarify that the DIPG36 is not wild type, it's G30.
Okay, G28, okay, yes, I'll fix that.
Yeah, just for everybody on the line.
Okay.
Yeah, this is with the glutamic acid, with the E mutation.
Okay. All right, so I think there's something with the combination. We'll continue, put all of that data set together and start coming up with some potential hypothesis for combos going forward. All right, thanks, Jerome. That looks good. Any other questions for Jerome? If not, let's move on to chemistry update. Who's next? Sue? Oh. Who is it? Okay, so Sue, you're up.
Yep, hi.
Okay, there we go. Enable anything. And then we go. Can you see that?
Yep, that's great. Thank you, Espen. Move on to the next slide. So over the previous month, we sent a couple of new compounds for screening. One of them was from our benzimidazole replacements for the trimethoxyphenyl, and one in the Blueprint series. We've also had the results from the earlier 5 compounds from the Blueprint series, which I shall go over. We've also had some new permeability data, which helps us to understand what sorts of modifications that we need to do to try and maintain the activity and enhance the brain penetration. And we're continuing with the synthesis in both of these series. So also, as Messin's mentioned, there was a new Blueprint patent, which we've gone through and identified a few compounds that look worthwhile to prepare. And also, as Messin mentioned, there's a new patent for Novartis, which provides some structures in it that may be of interest to as all other replacements for the trimethoxyphenyl. Go on to the next slide, please. So just to summarize where we're at with the benzimidazole, so this compound 3077 was a compound we'd identified previously, which has good ALK2 activity. We've lost some of the selectivity over ALK5 with this compound, but we had to. Well, in the original synthesis, we made it in a non-selective manner, and then had some difficulty separating the isomers where we've alkylated at the end. as we didn't know which one was going to be the best. This isomer was the better one. We've now prepared it by a more selective way where you can only get the one isomer, and we've sent a sample of that now to OICR so that they can put it through the AbbVie profiling in particular, looking at whether we can maintain some reasonable permeability with some of these compounds. We've also recently, the compound that we've synthesized and sent for screening is the corresponding analog, but with a nitrile in the 4 position of the pyridine. We're making some more compounds in this area where we're modifying the alkyl group on the benzimidazole. We're also preparing some compounds with where we're going to alkylate on the piperazine nitrogen, trying to modulate the lipophilicity of these molecules and trying to ensure that we get good cell permeability, etc. Move on to the next slide. Thank you. So this is just the introduction slide to the blueprint series. So this compound, obviously M4K2007, we know has got good activity, good selectivity. The one thing that it lacked was good permeability, and this was borne out when we did the PK study, when we found that the compound had poor brain penetration. And we looked at it and we thought that looking at the structure, it's got quite a high molecular weight, it's got moderate log P, but the pKa is quite high, and it actually it's a little bit variable. I think I might have used a different version in a later slide. But it's quite a high pKa. It depends which calculation method you use, but it's always high. So our plans were to look to see how we could modulate those physchem properties to make some compounds in this series that maintained that good potency, and also but had better physchem properties, so that we would try and enhance the brain penetration. So we use an MPO score, and most of the targets have at least an MPO score of about 4.5, preferably they're greater than 5, which means that they've got very high potential for penetrating into the brain. So move on to the next slide, please. So this was one of the compounds. So the original compound M4K3080 had moderate ALK2 potency, was very selective. I think this was replacing the core by the pyrrolotriazine. We were hoping that by incorporating the additional nitrogen in the phenyl ring on the right-hand side, enhanced potency, because we've seen from some of the modeling that this introduces an interaction with a tyrosine 285, which was observed in the crystal structure of M4K3007. Unfortunately, in this case, we didn't get the enhanced potency. In fact, there was no difference between the phenyl and the pyridyl. All you're doing is actually potentially probably making it a little less permeable with the higher TPSA. So it's a little bit disappointing. But if you move on to the next slide, please. So we also made a couple of compounds where we replaced the piperazine nitrogen in the top ring, the northern ring, by piperidine, or the unsaturated piperidine. Again, we were thinking that this would ensure that we'd removed that basic center there, reduced the basicity of the pyridine nitrogen. Unfortunately, these compounds were both inactive, and we've since sort of looked a little closer into this to try and understand why. And I think probably what we have done is we've reduced the basicity of that pyridine nitrogen too far, such that if you look at the two pictures in the bottom, we're looking at the electron density of around the pyridine nitrogen, which is going to interact at the hinge. And when you go to the carbon-linked compound on the right-hand side, you can see that the electron density is significantly lower around the pyridine nitrogen, making it less likely to be able to form that hydrogen bond at the hinge. So we're using this information now in our designs to just check a few of these things out, see whether or not we can still retain the potency with some of the modifications we want to make. So I guess we stay away from a carbon link compound was the bottom line of this. Unfortunately, we designed compounds around that, which was a little bit disappointing, but sorry. Did you have a question?
No, go ahead. No, go ahead. Yeah, sorry.
Move on to the next slide then, please. So this was a little bit more pleasing. So so if you take on the left-hand side, you've got M4K2007. So if you look down the physchem properties of this compound, it's got a molecular weight around over 500. pKa, as I said, this is for a different calculation, is around 10.2. It's somewhere between 9.5 and 10. And the result of this was it's got a relatively low MPO score, and the permeability was poor. And with a poor permeability and a high efflux or a moderate efflux ratio, when you get these determinations, it's unlikely to be getting into the brain. So all of the compounds from there across were designed to try and identify which of the features were key. So M4K3078 still retains that high molecular weight. It's still got a relatively high TPSA. It's got a lower log P. pKa has come down to around 8.99. And pleasingly, that compound has significantly improved permeability, and we've removed the efflux that we're seeing. The other three compounds have all got lower molecular weight. So key, I think, and they all have better permeability and efflux and no efflux. So I think the key message was it's not molecular weight that's driving the poor brain penetration. It's more likely to be the pKa. So we were quite pleased with this to see that we've managed to make compounds that have addressed the concerns that we had from 3007. And as Messlin mentioned earlier, 3078 and 3079 have been resized and being shipped off so that you can get a PK study carried out on these. Hopefully they will have good brain exposure.
Yeah.
Move on to the next slide, please. So we've moved away from the targets where we've got a carbon link on the top ring. So we're investigating an oxygen link compound. We've got a few others. There's a number of other targets where we're modifying the core or putting a substituent on the core, which I've discussed in previous meetings. But I think these are a couple of targets that now come into the frame where we've maintained either an oxygen or a nitrogen link, and these retain the higher electron density around the pyridazine nitrogen in order to ensure that we retain that interaction at the hinge. So these two targets actually will retain. We've done some modelling on these. I haven't shown the pictures here. They give the reasonable docking. They also have retained the acceptable physchem properties, so the pKa is in a reasonable area. Hopefully they will ensure that we don't get efflux. We're also looking at some other sort of combinations around these sorts of areas, which take us away from the original patent. And we'll also include some of these where the targets I've drawn up don't have the N-alkyl group, but we'll make those as well.
Okay.
Move on to the next slide, please. Sorry, Con. No, okay.
Hello? Oh, sorry. Yeah.
Sorry, I thought you were about to ask a question. Sorry.
No.
So this slide 9 shows the this second blueprint patent that came out probably about 6 weeks ago, I think it is. So it's similar to the previous patent. So you've got the piperazine. A lot of the compounds now, rather than having an amide on the top, actually have a carbamate. But the key difference really is they have an aza indole replacing the pyrrolopyridazine. Pyrrolopyridazine, sorry, the indazole thing. The generic structure shown there. Interestingly, they have quite a lot of compounds that are quite active, but don't have the whole extension in the solvent-based region. So there's a couple of the compounds. One of the compounds I've shown on the bottom right. We've docked this, and they dock pretty similarly to the original blueprint compounds. We wondered whether there might be a shift with it being a bidentate hinge interaction. But I think what happens is that the bidentate hinge interaction means that you don't need so much of the other interactions that are going on in the solvent exposed, going towards the solvent. We selected 3 compounds from this patent just to try and understand how these look in our assays, and these will be relatively straightforward to prepare. So we thought it was worthwhile preparing them. Interestingly, compound 68 that I've shown there has this nitrile, and I was interested to see whether that nitrile sits in the same position as our nitrile in compounds like 2106. And you can see from the docking picture on the right-hand side that indeed it does sit in a very similar position to the nitrile that we have. What we can't do, or it will be quite a challenge to do, is in the other series with the first blueprint series was to put a nitrile in that region. I looked to see whether that was synthetically accessible, but it really doesn't look that easy to make. So we're going to make these 3 compounds and see whether they show any advantage in terms of their properties, etc.
Okay, it's good. It's good ideas, yeah.
So if you move on to the last slide, I think it is. So this is the Novartis patent that Methvin mentioned, where they described that PD assay. So this is more similar to the our own pyridine series, where they've replaced our trimethoxyphenyl by this pyridone, N-alkyl pyridone. It's interesting, I looked back through the database, and actually we had made, or that it was amongst all the legacy compounds, I noticed a pyridone, but it was the other isomer. So the nitrogen was in where the carbonyl is, and the carbonyl was where the nitrogen is, interestingly. And that, well, I haven't got any activity for that compound, but I can give you the compound numbers if you're interested to see whether they had any activity.
Okay, yeah, interesting.
So in the patent, I think I've pulled out compound 1, which is one of the most active. It's the compound that they actually do most of the work on. And you can see what the activity is like. So they look at ALK2, ALK1, ALK5, and ALK6. It's interesting because a lot of compounds normally seem to have similar activity in ALK2 and ALK1. But we have got some selectivity here and some limited selectivity over ALK6. So I know David Smil said that he was going to look into perhaps making some of these types of compounds.
Yeah, we'll do like you suggested, the nitrile and the methyl versions of them to see if we get any more potency.
Yeah. Because they don't, in their example, in their patent, Y can be a nitrogen on the generic structure. Y can be a nitrogen or a CH or a CF.
Yeah.
So they don't include methyl or alkyl or C nitrile or anything like that. So that was interesting to see. They may be quite, the nitrile compound might be quite polar, of course.
Yes. We generally get about a 2-fold bump, 2 to 3-fold bump with the nitrile. So because these potencies are quite low. So that would be interesting to see.
Yeah.
Good.
Okay. So yeah, that's all I've got to say. Thank you.
Thank you. That's fantastic. No, not for me. This is great. Any other questions for Sue? If not, move on to Diva. Thanks, Sue. Yeah. Slide 33 enable. Starting toxicity. Okay.
So hi everyone, I hope everyone's doing well. The last presentation that I gave was around 2 months ago, and I've been discussing how we gained some interest in two amide compounds, specifically 2184 and 2192. So these compounds are potent inhibitors of ALK2, and they offer the additional advantage of having reduced affinity for the hERG channel. However, they have a less than desirable pharmacokinetic profile, as indicated with the table on the left. And when tested in the Keiko-2 permeability assay, these compounds had relatively high efflux ratio and less than ideal permeability from the apical to basolateral surface of the Keiko-2 monolayer. So there were a couple of ideas that we had to try to improve their permeability. Can you go to the next slide? So in the literature, it's been suggested that having these free N-to-H bonds sort of increases the chances of drug molecules to be recognized by efflux transporters. So we thought that capping that N-H bond with a variety of alkyl groups could potentially improve permeability and reduce recognition by these efflux transporters. So if you could go to the next slide. Before I actually give the Keiko-2 values for these compounds, I think it's important to see whether capping of that terminal piperazine nitrogen actually was detrimental to potency, and it wasn't. So in the RBC biochemical kinase assay, the potency was actually improved slightly, and selectivity over ALK5 also increased, which was very good. Unfortunately, this didn't really translate to an improvement in cell-based permeability. It was still within 80 to 100 nanomolars, but we still were able to maintain a favorable hERG profile for all of these analogs. For the dimethoxyamide compounds, potency was maintained. There wasn't much of an improvement in terms of selectivity. We found that these compounds do have a good potency for ALK5, so it's a little bit difficult to sort of play around with the selectivity. But once again, hERG profile was still very good. You had an IC50 value of greater than 30 micromolar for all the analogs. Moving on to Keiko-2, so there was a significant reduction in the efflux ratio for the fluoromethoxyamide compounds. We'd gone from about 40 to between 2 to 3, which was great. So this is sort of like a proof of concept. It would be better if the efflux ratio was closer to 1, but we can also see that the permeability of these compounds was increased as well. So that was definitely a good thing. If you can go to the next slide. For the dimethoxyamide compounds, unfortunately, the efflux ratio is still very high. It was greater than 10. There wasn't much of an increase in the permeability, so these compounds are not as promising as we had initially anticipated. So there's not much that we can do for that. And now looking at the metabolic stability of these compounds in liver microsomal stability assays, the compounds are comparable to 2009. 2234 had pretty good stability in both MLM and HLM, so that was quite good as well. The next slide. So for the dimethoxyamide compounds, it was the same thing. That 126 trimethylpiperazine motif seems to be a little less stable than the other alkyl group. There was also a rapid PK experiment that had been done for 3 out of the 6 analogs that we had made. So I've provided reference values for 2009. So these are just rough values. As you can see for 2239, the plasma concentrations are not that high at a 25 mg/kg dose in mice. The values are a little bit better for 2236 and 2238, but it seems that the highest plasma concentration was achieved for 2009 at approximately 6 hours after the dosing, whereas for M4K2236 it's approximately 4,000 nanomolars at 30 minutes, and it seems like there's a gradual decline in plasma concentration after 30 minutes for these analogs. And just a few other analogs that were made in the past couple of months. So replacement of the paramethoxy group for a nitrile showed good potency in the biochemical kinase assay, but unfortunately this didn't translate to a really good potency nanobret assay. And as I highlighted in the previous presentation, having that primary amide in the pair position of the arrow ring is crucial for maintaining potency. So for 2240 and 2244, there was a significant reduction in potency. And then finally, an analog of 2192 was 2250. So we were trying to sort of improve the permeability of these dimethoxyamide compounds by installing a nitrile group at the 4 position of the pyridine to try to attenuate the basicity of the pyridine. And I have the Keiko-2 values for that compound in the next slide. Unfortunately, this didn't do much either. The efflux ratio was still quite high, and permeability was less than ideal as well. You can go to the next one. And finally, we had noticed for a couple of other amide analogs that methyl group on the 4 position of the pyridine ring was actually not doing much in terms of potency and selectivity. So I decided to test 2 new analogs, 2251, 2252, without the methyl. And we can see for the fluoromethoxyamide, there is a bit of an improvement in selectivity and even an improvement in potency. For 2252, there was a slight improvement in potency and just a slight increase in selectivity, but this may be negligible. So, yeah, that's it.
That's good. It's interesting. Interesting to do the analytical versions of that more potent system to see. Yeah, whether or not that methyl that we put in on the pyridine was a liability. Yeah. Okay, any other questions for Diva? Thank you. That was good. Good update. Okay, if not, okay, thanks guys for giving us this in-depth chemistry overview. The next thing we're going to do now is get some update from Owen and Arish on the response from the clinicians about ALK2 and about going towards the clinic with a candidate. It's an ALK2 inhibitor. Yeah.
Okay, so hello everyone. So on May 22nd and May 31st, M4K held an in-person and phone meetings with pediatric neuro-oncologists who have experience with clinical trials and treatment of gliomas, including DIPG. There was a clear and urgent, sorry, next slide, please.
Oops, sorry.
It was clear from the discussions that there was an urgent need for DIPG treatment options. Owen and Mesfin presented the progress to date on the lead compound for M4K, the M4K2009, including the FOP mouse model work that was completed by Dr. Eileen Shore and the combination studies with other inhibitors in DIPG cell lines. We received helpful suggestions on follow-on studies to complete. There was general consensus that the compounds would be dosed in combination, so it would be helpful to determine combination effects with other inhibitors and chemotherapy. We also consulted the clinical advisors on possible treatment protocols during clinical trials. The standard of care is radiation, and it's usually given prior to administering a trial drug, unless there's preclinical evidence to suggest that the drug is radiosensitive, in which case it would be given alongside radiation. Biopsies are not routine, and so they cannot be added as part of a protocol, so it might not be possible for us to figure out which patients have the ACVR1 mutation beforehand. Biopsies are only done if there's an atypical tumor and there's a question about the diagnosis. Next slide, please. We also learned that there are clinical trial consortia that could help with funding and patient recruitment, such as the PNOC consortia, which is based in the US, as well as the CONNECT consortia, which has sites in North America, Europe, and Australia. The advisors also suggested strongly that we consult with regulatory advisors to help determine the minimum required efficacy in our in vivo models to help satisfy the clinicians, make them comfortable about using our our compound in their patients, as well as to satisfy the institutional review boards. In general, there was broad support to move the program forward, which was great news for us. And we heard a lot of different opinions and feedback, which we can use in designing our next stage. I'd like to take this opportunity to thank our clinical advisors for their time, as well as their invaluable input to help us in the next round of studies.
That's good. Just very quickly, I think just one of the biggest takeaways from our side is, besides the dire need for a cure, as we go forward in our next session, we're really going to have to bring in some regulatory bodies, because the regulatory thoughts on this are moving, and so that's going to be a key part to all this. Yeah. So.
It's good. Thanks, guys. Any other questions from Owen and Naresh and the clinical update? If not, I think that was most for presentation updates for today's meeting. So like I say, we have identified a few candidates from the PVD lead series. We have identified a few compounds from the blueprint backup series. We're going to continue to characterize those. In about a week and a half, 2 weeks, we should probably have gathered all of the data we need from the lead series to kind of see if we can whittle down that list of 6, 7 compounds down to a decent 4 or 5 that we can start getting some more toxicology type of information on and start thinking about scaling some of those up. Well, like I say, everything is looking promising. I mean, so we have 209 that has nice properties, and we have analogs very similar to those with the hERG liability, and now we are starting to identify compounds that gets rid of that hERG liability. Hopefully they preserve all of the nice properties of 2009. So we're in a very good place. I think very, very soon we should be able to declare some candidates for the end of this first phase of the program. But like Owen said, I mean, over the next year or so, we're going to identify other compounds that we can scale up. We're going to get tox information and start thinking about more advanced studies, preclinical studies that we might want to do. Okay, anything else?
So when you nail down to the 4 compounds or whatever, how much material would you want to scale up, and would that be done in-house with chemistry here?
Most likely we're actually going to use the CRO. I mean, for 2009, I mean, it was nice that GL chimed in and deliver quantities that afford us to now to be able to do a proof of principle. We might reach out to GL again, but we might shop around for other sources. Yeah, we
will definitely go back to GL personally and be really supportive, and we want to, thank them very much for what they've done in the past. Yeah, at our next meeting, we will pick sometime in the next 2 weeks. Yeah. I think our next meeting is July 3rd.
Yes,
We'll present what we think are our candidates on July 3rd. There will be kind of a chance for people to chime in or, quiz why we picked what we did. But we're going to have, because of the timelines, we're probably going to have to be moving forward pretty rapidly. And frankly, I think we're starting to narrow down. I think the data is driving what the answer is going to be. There may be 1 or 2 on the outside, but I think the core 3 or 4 are kind of rising to the top right now. But the final data we get over the next 2 weeks will really determine that. So
that's good. The other thing about so maybe just to update, so for the in vivo models, I mean, we are engaging Chris Jones and Angel. They look like they're both interested in wanting to plan an in vivo efficacy study with the DIPG 007 line. I mean, like I've always said, one of the nice things about 2009 is it looks like we can dose it high, so hopefully we'll be able to show some nice efficacy in their model. But I mean, if other people are willing to put it in vivo and test in other models, please let us know. We don't have a shortage of compounds. Yeah, so keep that in mind.
Otherwise, our next meeting is same time on July 3rd.
Yes.
So thanks everyone for everything.